So our last speaker today, I would like to introduce Dr. Ben Liesfeld. And after obtaining his PhD in physics, he dedicated himself to the development of computer-guided laser surgery devices for a startup for ophthalmological medical devices. Afterwards, he worked at the clinical laboratory for genetic diagnostics, where he was responsible for establishing IT software development and the bioinformatics department. This is where he also learned about the huge potential, but also challenges of clinical genetics. And this led him to found his own business. Now, since 2014, he's the co-founder and managing director of Limbus Medical Technologies. And today, he will talk about why performance matters in the long-read data analysis in medical device software. Welcome, Ben.
Yeah, thank you, Yvonne, for the kind introduction. I'm really excited to be back in Milan. And I'm also very excited to see so many interesting sessions about long-read sequencing, about new insights that we can gain from long-read sequencing. I think this topic is really picking up. And while I get easily excited about new technology, I'm a physicist by training, I'm also responsible for translating new technology into a clinical setting. So what do we have to do? What do we have to take care of in order to use new technology in a patient setting safely? So that's where performance matters in many different aspects. We, as a software development company, when we think about what a medical device software should deliver for long-read sequencing, we think mainly of three different things. First of all, we think of ease of use. When you're in routine workflow, you need to be quick. Software needs to be usable very easily. This for us means hands-free use. We like to automate as much as possible, and we like to translate our short-read sequencing workflow, which starts from raw sequencing data and ends with interpretation of data. We translate this into long-read sequencing as well. We also emphasize that it's very important to have all information in one place, not only just short-read sequencing and long-read sequencing data, but also all the information that you need to interpret the variants. And last but not least, when you look at your data, you need to have a good understanding of how accurate it is, how reliable it is, how complete your assay is. And this is also something that is our responsibility as a medical device manufacturer. Just a short review, we are supporting a number of sequencing technologies. We've started with Illumina-based technology, MGI, Element Biosciences, short-read technology. Since January, we also support long-read sequencing data from PacBio. And since many people have asked us about this, of course, we will be supporting Oxford Nanopore as well in the near future. So stay tuned about news about this as well. There are certain applications that have not been accessible to us using short-read sequencing data. And everyone in the room who has been thinking about long-read is probably very familiar with those shortcomings. There are these so-called dark regions, which we simply cannot access very well in the genome. This affects also other analysis types that we typically provide for short-read sequencing, like CNV analysis. Repeat expansions have been very difficult, typically, or in particular when they are large expansions related to pathogenic variants. And of course, structural variant calling has been kind of a gamble with short reads, and this is being promised to be so much better with long read sequencing data. If you need another reason, and the text is cut off here a little bit, if you need another reason to switch to HD38, I know that still many clinical laboratories are using HD19, so we will only be supporting long read sequencing for HD38 for a number of reasons. And of course, we would like to migrate to T2T in the short future, but there's a lot of things that have to come together to make that possible in a clinical setting. When we speak about ease of use, we also speak about a very simple IT architecture that is required on a customer side. So when you're using PacBio technology, you typically only need the instrument and maybe a small computer infrastructure to operate SmartLink software. But once data has been pre-processed on the review device, it can be already uploaded into the varvis cloud and processed there. If you look a little deeper on the process level, what has to happen on the device is the consensus read calculation. If you're using third-party indexes, third-party assays on the review device, you need to perform an additional step for demultiplexing, but kind of the underlined raw reads can be uploaded to the varvis software and processed from there. I'd like to give you two examples where convenient visualization and the comprehensive view of all the data that you have about a patient facilitates the interpretation. So here is an example of combining our conventional CNV analysis with the additional high-quality structural variant analysis in our software. And this is kind of the regular view that you typically have in this for the CNV analysis. At the bottom of the screen, you can see a tabular representation of all the structural variants that were found in the sample. And above that table, you see an IGV visualization. And the two important tracks here are at the top. So the red bar is the result from the CNV analysis, and below that, the blue bar is from the structural variant analysis. And here they match nicely together. There may be cases where systematically you cannot detect structural variants, for example, in targeted enrichment assays. There, the CNV analysis may complement your findings, and vice versa. We provide a number of details about the variants, annotations about those structural variants. We perform lookups for you in population databases like Decipher and DGV, and also GNOMAD. So that annotations that help you interpreting the structural variant. And if you have a larger screen, you can also add a view of the read alignment. And you can see here nicely that the structural variant called the blue bar actually represents the structural variant more accurately, because you can accurately see the breakpoints here. Another example of how good visualization and comprehensive data view supports the analysis is tandem repeat expansions. We put a lot of thought into how to display and present those in our software. Again, for the quick view, we provide a tabular overview, and you can quickly filter to the variants that are of interest to you, that are deviating, that are exceeding certain thresholds. But we also learned a lot about how interruptions, how the actual structure of repeat expansions can impact the interpretation. So it's important to know how these motifs are, where the motifs are located, and if they are interrupted. So this example here, the pathogenic repeat is in orange, and the gray bars in between are interruptions that may impact the pathogenicity of the variant. And we provide other context information. We show you how the length of the specific alleles are related to certain thresholds for intermediate or pathogenic status. We show you information about age of onset and other information from the literature, so that you have a complete picture and all the information available to enable the classification of this variant. I'd like to highlight a very important resource here and important work from Harriet Deschnau. She's maintaining ST Archive, a comprehensive resource of known repeat expansions or known pathogenic repeat expansions, contains now 74 loci. And she does a tremendous work consolidating knowledge about those tandem repeats, but she also provides a platform to gather feedback, because there's so much information now flowing in, new loci being examined, and there's a very healthy discussion about how they should be interpreted. So this is very important work that she's doing. I also like to highlight that there's now tandem repeat information available from GNOMAD. Just keep in mind, this data has been generated using short read, whole genome sequencing data analyzed with Expansion Hunter. And this is different quality of information, of course, different quality of data, but still it gives you kind of an outlook of the data that we can now generate using the varvis software when you comprehensively examine all tandem repeats in all your patients. And this display here shows you a two-dimensional graph. And the information that we collect is two-dimensional. You have a longer allele on the x-axis and the short allele on the y-axis. And you can see that, obviously this is a recessive disease. You can see that a large number of individuals carry expanded alleles. And when you highlight the thresholds for intermediate or pathogenic status, this nicely matches kind of the statistics that you get from a healthy population. And we will learn a lot more now when we perform this analysis on all our patients. There's so much new discoveries to be made also in loci that we cannot interpret correctly right now. So this again will support analysis in our software. And let me quickly touch about on analytical performance. When you look at data, you need to be sure of what it means, what the limitations of your assay are, as any assay has limitations. And this is an image of myself in younger years when I had dark hair still, and I was lamenting about the lack of reference material. When you have a new method that is so much better than all the legacy methods, how do you really prove how much better it is if you don't have kind of the comparison? And fear not, Genome in a Bottle project, in conjunction with device manufacturers, has put a lot of work into characterizing reference material. They took HG002 and accurately analyzed more than 1 million different repeat loci in this genome. And while this genome this sample is a healthy individual, it still provides a very useful reference. So since it's a healthy individual, it only contains very few expanded repeats. You have to take that, or keep that in mind and take that into consideration when really benchmarking. But it's really useful when comparing methods and also comparing the progress that you make in development, changes in your assay. And you can also, and I'll give you a few examples. This is just a graphical representation. You can see that that the number of longer repeats decreases in this human genome. So the number of really kind of those expanded repeats that you are interested in a patient and in pathogenic variants, this is underrepresented here, and you need to correct for that. So when you use this reference material and do different methods, for example, you can compare short-read tandem repeat analysis with long-read tandem repeat analysis. You can see here that specificity is high in both methods, but sensitivity differs a lot. So you can detect with higher sensitivity expansions using long-read sequencing. But you have to be careful. Actually, kind of on the right side, we have the total number of tandem repeats that are accessible to short-read sequencing is much smaller than the number of repeat expansions that are accessible to long-read sequencing. You have actually to exclude a number of complex loci because they're not accessible to short-read sequencing. So it's not only higher sensitivity, but you gain access to so many more regions in the genome using long-read sequencing. Just as a summary, when you're looking at different solutions in the field on this conference, things you have to watch out for in tandem repeat analysis. First of all, and that's something that I learned in the past months, it's very important how you define repeats. What is actually the location of the repeat? Is it a single repeat location, or is it a cluster of repeats? This impacts how you analyze this in a bioinformatics way, but it also impacts how you can interpret it clinically. You have to take care of issues with false negatives. The length of your reads in your assay are still a limitation. No matter what technology you're looking at, of course it's a limitation in short-read sequencing, but it may also be limitation in long-read sequencing. And the motives can become complex. This can also impact the analysis. And last but not least, it's these interruptions that seem to be very interesting. We have not been able to discover them using legacy methods. We are now being able to see them with long-read sequencing, and it's very important that we capture that information and can together make sense of them. So just an overview of what we are planning in our software. I told you from January already we are supporting long-read sequencing, small variant calling and phasing. And from our next release, version 2.6.0 after ESHG, we will also be supporting CNV analysis, repeat expansion analysis, and structural variants in our software. And if you'd like to see more details, we would be happy to give you a demo at our booth. And thank you for your attention.
Yeah, so now we have 15 minutes still for Q&A. So if there are some questions, feel free to raise them now. There is one question. Yes, please.
First of all, thank you for your beautiful presentations. I don't have any specific question about your presentations, but I have a question about varvis. After the detection of the variant, what about the process of the interpretation? Does varvis have any commercial version of the variant interpretations for the other clinical geneticists?
So varvis is a commercial software, and you basically the business model is you pay for the entire analysis once. You can update annotations at any time. That's not limited. So the analysis includes everything, both the bioinformatics as well as the interpretation.
Actually, I mean free use version on the websites or on internet, like Varsam or Franklin.
We don't have a free version. It's a medical device. Exactly.
What do you think about that? Should varvis have to or not? Because I think it's at some point useful for the company also to create a database.
I agree. I mean, free tools have value, and it's just a matter of resources, what we can spend them on. And long-read sequencing, now this kind of solution for the patient has been our focus.
Thank you.
If you're wondering about a trial possibility, this is possible with varvis. You can get in touch with us, and then we can help you with getting a free trial.
Okay, so here's another question.
Yeah, hi. What about the methylation detection and translocations?
Yeah, methylation is also something that we are very interested in. In the tandem repeat analysis, we're already providing summary information about methylation, and this will become more detailed in the near future. I've just been at London Calling, the Oxford Nanopore conference last week, and there's a lot that you can learn from methylation. So yes, will be integrated. Probably not, only as summary information in the next software version, and will be expanded in the future. And translocations, more complex structural variation, are being called by the structural variant caller. And from my point of view, the issue is how do you visualize, how do you make sense of those complex variations? And also this is ongoing. So we provide you some, let's say, basic visualization right now and annotations with that. And we have a number of ideas how to improve that. But we're also here to interact with researchers, with clinicians. How would you like to see that information, right? What is the information, what's the visual cue that you need to interpret the variant?
A circus plot would be nice. Visualize translocations and circus plot.
Yeah, it's our top number one priority.
Quick question. So about like short reads comparing to long reads, because a lot of GWAS is still like single variant focused. I'm wondering, how do you think the accuracy comparing short read sequencing to long read sequencing? Are they like short reads are have more accuracy over like SNPs?
Well, the thing is, with short read sequencing, you have the community has accepted that we cannot make variant calls in certain regions at all, right? So we're excluding those regions.
I see.
And now we can access them with long read. So it's, I think, like in the regular regions, the difference is not that great,
much.
right? It's not that much, but of course you have access now to difficult regions that you did not know about before at all. And that's for the small variants, and for the, of course, for the longer, for the structural variants, there's a great benefit of.
Yes. Thank you.
So no further questions at this point, but maybe you have questions later. Feel free to contact us, visit the booth, or contact us later by email on the website. And with this, I would like to conclude. Thank you all, and don't forget, read long and prosper. See you around. Bye.